Okay, so welcome to this next video on mitosis. Okay, so far what we have done is we've got to metaphase of the mitosis phase of the cell cycle. Now, in metaphase, what happens is all of the sister chromatids align down the center of the cell, down the axis of division, basically. What follows is a very rigorous checkpoint to make sure that every single sister chromatid has been linked to a mitotic spindle, and to make sure that every spindle pole has attached to a sister chromatid of all 46 chromosomes within the cell. Okay, this is the spindle assembly checkpoint, and basically the way it works is that if a kinetochore has not had a mitotic spindle bound to it, then it will bind this mitotic checkpoint complex, which will sequester the CDC20 protein. And when CDC20 is sequestered, it will not activate the anaphase promoting complex / cyclosome. So, in order to get activation of the anaphase promoting complex / cyclosome, what you have to have is you have to have all of the mitotic spindles bound to kinetochores. So you have to have every single kinetochore with a mitotic spindle attached to it. That displaces the mitotic checkpoint complex, and when the mitotic checkpoint complex is displaced, it can no longer sequester CDC20. So CDC20 is released, and it goes off and activates the anaphase promoting complex / cyclosome. Okay, now the anaphase promoting complex / cyclosome is then responsible for pushing the cell into anaphase. Okay, and in anaphase, what's going to happen is that the mitotic spindles are going to pull the sister chromatids apart, basically. Okay, so what's going to happen is that you're going to pull, and oh dear, I'm doing it in color. Right, so here are these mitotic spindles, like so, and I'm running out of this blue pen, so I'll have to be economic with the number that I draw. In fact, no, I want to keep it the same. 5 of them, here we go. Of course, there will really be 46 of these, but never mind. So they've pulled, basically, these sister chromatids apart, and now these structures that they have here are the actual chromosomes, or the single chromatids. Those are a single copy of the DNA. They're no longer two of the identical copies stuck together, so you've pulled the two sister chromatids apart, and this basically is anaphase of the mitosis. So in anaphase, you pull the two sister chromatids apart, and they go towards these spindle poles, which are at either side of the cell, basically. Okay, and in this way you're getting ready, basically, to form two separate nuclei, each with the full genome of the cell. These are the spindle poles. Right, okay, now how does the anaphase promoting complex / cyclosome trigger this to happen? Well, basically what it does is it activates an enzyme known as separase. Okay, so let's have this enzyme separase here. Right, and basically this separase enzyme is usually inhibited by a subunit known as securin. So it usually has bound to it a protein known as securin. So this is securin. Okay, and I will colour that in a certain colour. So we'll have securin in orange. Let's have securin in orange. So securin is usually bound to this enzyme separase, and if it's bound to separase, it basically inhibits separase from functioning. Now, the anaphase promoting complex / cyclosome is going to work by cleaving securin off separase, and then when you've got separase on its own without securin, it basically becomes active. So here is the active separase, which has no securin inhibiting it anymore. Now, what does separase do? Well, basically, do you remember me telling you that between the two sister chromatids, you had a huge number of proteins? So you had a huge number of connections between these two sister chromatids, which were holding them together. And the name of these proteins were cohesins. These were cohesins. Okay? Well, basically, what separase does is it goes over and breaks all these cohesins between the two sister chromatids, and that now allows the two sister chromatids to be pulled apart, basically. So this breaks down cohesins, okay, and that allows the two sister chromatids to be pulled apart. Breaks down cohesins. Right, okay, so that's how the anaphase promoting complex slash cyclosome promotes the movement into apoptosis and the splitting of the sister chromatids. Right, it also has another function. It wants to ensure that you don't go through mitosis again. So it now needs to inactivate the MCDK, okay? So the MCDK, remember, this cyclin B, and I'll draw the inhibit. So MCDK was cyclin B CDK1 complexes, and it was also called the MCDK. Okay, and this enzyme was important in making the chromosomes condense. It was important in forming the mitotic spindles. It was important in allowing the mitotic spindles to capture chromosomes and important in making the nuclear envelope break down. It was very, very important for the early stages of mitosis. You want to stop all of that stuff happening now because you want to reform the nuclear envelope around these two new genomes, basically. You want to form two nuclei, so you need to inhibit this enzyme now. And the way it's done is by the anaphase promoting complex. So basically, remember that cyclin B CDK1 complexes consist of these two proteins, namely cyclin B and CDK1. So this here is CDK1, and this here is cyclin B. Okay, right. And the way in which these anaphase promoting complex, I'm just going to colour in these CDK1 cyclin B complexes. The way in which the anaphase promoting complex prevents the activity of these cyclin B CDK1 complexes is by targeting the cyclin B for destruction, basically. So it's going to remove the cyclin B, and without the cyclin B, this can't exist. I mean, okay, yes, it has to have the phosphate group on to activate it, but the cyclin B is rather essential. So basically, what this enzyme is going to do is it's going to work with two other enzymes, which are called E1 and E2, and together what they're going to do is they're going to stick multiple ubiquitin groups onto this protein. So you're going to get a polyubiquitin tail here. So they're going to stick these ubiquitin groups onto the cyclin B. And proteins which get ubiquitin stuck onto them then get destroyed by the proteasome. So here's the proteasome. It's going to take the cyclin B in, and it's going to break it down into its amino acid components. So, in this way, cyclin B is going to be degraded when the anaphase promoting complex or cyclosome is active. So, by breaking down that cyclin B, you're going to inactivate these cyclin B CDK1 complexes. Okay, so that's how you stop the early processes of mitosis from happening again, basically.